Purin synthesis is very difficult, so we're going to show the easiest way to understand it. We begin with 5-phosphoribosyl 1 pyrophosphate and diglutamine, which has a carboxyl group and a alpha carbon, a amino group, and a hydrogen, and in the R group has 2 CH2 and has a carboxyl group, but instead of here having the OH has a amino group, and this amino is going to be attached here in the first carbon of the ribose and release this pyrophosphate. Then the glycine is going to, well, the glycine is the group carboxyl, the alpha carbon, the amino group, and in the R group is a hydrogen, to the carboxyl group. And it is going to join to this amino group and is going to release this water. Then the formyl tetrahydrofolate donates a group, formyl group, and then another glutamine comes again, and the glutamine is the carboxyl group, the alpha carbon, the amino group, two carbons each with two hydrogens, and a carbon with a oxygen and a amino group, and this amino group is going to join to this carbon and is going to release water. Then, in humans, the enzyme takes carbon dioxide, but in bacteria, the enzyme takes bicarbonate. First, an enzyme puts a double bond here, and then another enzyme takes the carbon dioxide and put it here. And then the aspartate, which is the alpha carbon. Here is the alpha carbon, the carboxyl group, the amino group, the hydrogen, and in the R group is a carbon with 2 hydrogens and a carboxyl group. This amino group is going to join to this carbon and it is released fumarate, which is a molecule from Krebs cycle, and then a carbon from formyl tetrahydrofolate is attached here in this amino group. And then the ring closes and these double bonds change. And so now we have the rings of the Purin. We never close that. We never close that ring. So now here are the two rings of the purines, and this is inosine, and the base, just this without the ribose, is hypoxanthine, and then the aspartate, donates the amino group and the aspartate has a alpha carbon, a hydrogen, a carboxyl group, the amino group, one CH2 and a carboxyl group, and this amino is going to attach here in this carbon. And so now the base, instead of being hypoxanthine, is adenine, and with the ribose is AMP. So this amino group came from the aspartate. Or instead of take aspartate, first a dehydrogenase takes here in the position 2 of the rings of the purine, puts a double bond with a oxygen, and then the glutamine, which is the glutamine that is the carboxyl group, the alpha carbon, the amino group, the hydrogen, 2 carbons, each with 2 hydrogens, and a carbon with a oxygen and a amino group. Now this amino group is going to be attached here instead of the oxygen. And so now we have GMP, which is guanosine monophosphate. So in the adenine, the amino group, which is here in the in the 6th position, because this is the first position, second first position, second position, third position, 4, 5, 6. So the amino which is in the sixth position of the adenine came from the amino group of the aspartate. And in the guanosine, the amino group that is in the second position came from the glutamine. And this is the seventh position, this is the eighth position, this is the ninth position, and this, the important positions is the sixth, because here goes the amino group of the adenine, the 9, because here here joins with the ribose. And the second position, because here is the amino group of the guanosine, and in the ribose, the carbons that are important is the 1, because the first carbon is joined to the base. In the second, it is decided if the ribose is ribose or deoxyribose. In the third, here joins the phosphate of another nucleotide in DNA synthesis or RNA, and the 5 because have this phosphate, and the 4 because it's attached, the 5 because the 4 because it's attached to a fifth carbon. We see you in the next metabolic pathway.